All right, today we're diving into what is basically a scientific detective story. The crime scene isn't some dark alley, though. It's poultry farms. And the victim? Well, it's one of the world's most important food sources, chicken. So here's the big question we're trying to answer. What is the biggest hidden threat to Malaysia's poultry industry? Because, as a group of scientists found out, the answer, well, it's not what anyone expected. You know, in any good mystery, you've got to have your suspects. And here are our two main culprits. First up, we've got Mycoplasma Gallisepticum, or MG. This is the notorious one, the usual suspect, known for causing really nasty respiratory disease and huge financial losses. And then there's Mycoplasma Synoviae, or MS. This one's more of a stealth operator. It often causes milder infections that can kind of fly under the radar. For years, MG has been public enemy number one. All right, so let's set the scene. Our story really kicks off with this sickness spreading through the flocks, a problem that, believe me, plagues poultry farmers all over the world. The disease itself is called avian mycoplasmosis. It's really a blanket term for the infections caused by our two suspects, MG and MS. Think about it. Breathing problems, swollen joints, and if you're laying hen, you're not producing as many eggs. For the farmers, this all adds up to major economic losses, and it affects everything from the well-being of the animals right down to the price of chicken you see at the supermarket. So, to get to the bottom of all this, a team of researchers in Malaysia launched a full-on investigation using modern science to hunt down the real culprit. And they went about it like true detectives. First up, they had to gather evidence. They collected 407 swab samples from chickens all over the region. Then they took those samples back to the lab to grow any bacteria that might be hiding in there. And finally, the really crucial part, they used a powerful technique called PCR, which is kind of like getting a unique DNA fingerprint for any bacteria to pinpoint exactly which of our suspects was at the scene of the crime. And wow, this right here, this is the smoking gun. This gel image is basically the scientific version of a police lineup. Each one of those bright bands is a positive ID. The lower band, that's a perfect match for our notorious suspect, MG. And that upper band, that's the DNA fingerprint for the stealthy one, MS. The evidence was crystal clear, but the pattern that emerged, that was the real surprise. And this, my friends, is where the plot takes a major twist. The data started pointing to a completely unexpected culprit, revealing that the main bad guy wasn't who everyone thought it was. The results, honestly, they were a total shocker. Just look at this breakdown. The stealthy one, MS, was found in a whopping 42.5% of the samples. Meanwhile, the notorious MG, the prime suspect for years, only showed up in about a quarter of the cases. The lesser-known bacteria was actually the dominant threat out there. You know, even the researchers themselves were taken aback by this. As they put it in their paper, these findings all seem to suggest the growing relevance of MS in the poultry industry. In other words, the quiet culprit was suddenly making a whole lot of noise in the data. But identifying the primary culprit was only half the mystery. The investigation uncovered a second and, frankly, much more alarming development, a growing resistance. And this leads us to an even more critical question. We've identified the main culprit, sure, but is it leveling up? Is this stealthy bacteria developing new powers that make it even harder to fight? And the answer, unfortunately, is a big yes. The scientists tested several common antibiotics against the MS bacteria they'd found. What you're seeing here is the result. Some drugs, like tivolosin and tiamulin, they're still working. But others, like erythromycin and lincomycin, are losing their punch. It's like the bacteria is literally learning to put on armor to fight off our best weapons. And the hard data? Well, it really paints a scary picture. For some of these MS strains, it took a dose as high as 64 micrograms per milliliter of certain antibiotics just to stop them from growing. Now, to give you an idea of how crazy high that is, the good antibiotics usually work at numbers way, way lower, like under 1 or 2. These are dangerously high numbers, and it signals serious resistance. Now, there's a scientific term for this measurement. It's called the Minimum Inhibitory Concentration, or MIC for short. You can think of it as finding the bacteria's breaking point, the absolute smallest dose needed to stop it in its tracks. So when you see that MIC number climbing higher and higher, yeah, that's a huge red flag. It means the drugs are losing the fight, and the bacteria is winning. So the investigation is complete. The evidence has all been analyzed. Now for the most important part, the verdict, and what this all really means for us. So let's break down the final verdict. Case closed. The prime suspect has totally shifted from MG to the once overlooked MS. And even more alarming, this new culprit is arming itself, building up resistance to common antibiotics. The bottom line? Farmers can't just guess anymore. Treatment has to be targeted and based on actual lab tests. And for now, it looks like tivolacin and tiamulin are the best lines of defense.
And here's the thing. You might be thinking, okay, this is about chickens. Why should I care? Well, this is so much bigger than that. This case in Malaysia isn't just a local story. It's a perfect snapshot of a terrifying global problem, the rise of antimicrobial resistance. The same kind of evolution that's happening on these farms is happening in bacteria that infect humans, threatening to make our modern medicines totally useless.
So this investigation solved one local mystery. But in doing that, it uncovered a much, much bigger threat. And it leaves us with one final, pretty sobering question. If bacteria on the farm are evolving this quickly, are we truly prepared for what comes next?